In this unit, we will be discussing various aspects of cell division. During cell division, a single parental cell divides into two daughter cells. This unit will be divided into 4 sections. The first involves the asexual replication processes of binary fission and mitosis. The second will discuss the details of meiosis and how it compares to mitosis. The third will examine non-disjunction and what happens when meiosis goes wrong. And the fourth will discuss the importance of cell cycle regulation and its role in cancer development. Now let's start with a discussion of mitosis. Cell division is the process by which cells make more cells. The process of cell division must satisfy several important requirements. The first is that the daughter cells must receive the full set of genetic material present in the single parent cell. This is accomplished by DNA replication, which will be discussed in Chapter 12. The second is that the parent cell must be large enough to divide in two and still contribute sufficient cytoplasmic components to each daughter cell. In prokaryotic cells, cell division occurs through binary fission. In eukaryotic cells, it occurs through mitosis and cytokinesis. During binary fission, a cell replicates its DNA, increases in size, and divides into two daughter cells. Remember that prokaryotes do not have a nucleus, so this process is pretty straightforward. The circular DNA that makes up the prokaryotic genome is attached to the cell membrane by a protein. Next, DNA replication is initiated at a specific location on the DNA molecule and proceeds in opposite directions around the circle. The two copies of the genome are both attached to the membrane with proteins. As the cell begins to elongate, this pulls the two circular genomes away from each other as their proteins move further away. New membrane and cell wall then begins to be built as the middle of the cell constricts, eventually forming two separate cells. Cell division in eukaryotes proceeds through a number of steps that make up the cell cycle. This is more complicated than binary fission due to the presence of the cell nucleus. The cell cycle has two key stages: the M phase, shown here in red, where the parent cell divides, and interphase, shown here in blue, where the cell is getting ready to divide. Interphase consists of 3 main stages: G1, S, and G2. G1 and G2 is where cell growth is occurring. In these stages, the cell physically gets bigger and makes more organelles, such as mitochondria, to distribute to the two daughter cells after division. During S phase, the S stands for synthesis, because this is where the DNA genome is replicated. Cells that are not undergoing the cell cycle are said to be in G0, so cannot divide to make new cells. This is why if you sever the spinal cord, the cells cannot replicate to make the connection. But if you cut your skin, your epithelial cells can make more cells to heal the cut. Your epithelial cells can leave G0 and enter into interphase. However, your nerve cells are permanently in G0. Because the genome must replicate and separate during cell division, it is important to understand the general structure of genomic DNA. In eukaryotic cell, DNA and protein work together to form chromatin, which are packaged into structures we call chromosomes. Each of your cells contains 2 full sets of chromosomes that make up the human genome. One set you got from your mother, and the other from your father during fertilization. The full set of a human genome contains 23 chromosomes. So each one of your cells contains a total of 46 chromosomes. They can be pictured in something called a karyotype, which is seen here. Cells with 2 sets of chromosomes are called diploid, and the chromosomes that carry the same set of genes, one from the mother and one from the father, are called homologous chromosomes. A cell with only 1 set of chromosomes is called haploid. Each one of the 46 chromosomes in the human genome must replicate during S phase before they can be passed on to the daughter cells. The 2 identical copies, called sister chromatids, do not completely separate, however. Instead, they stay attached to each other at the centromere. During mitosis, the sister chromatids must be separated from each other so that both daughter cells will obtain a complete copy of the genome. There are 5 key stages of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. It is important that you understand the key cellular changes that occur at each stage. You can find that information in your textbook. In this video, we're going to focus on what occurs to the movement of the chromosomes in each stage of mitosis. I'm going to use a cell to diagram the stages of mitosis. Here's our nucleus, and this cell is going to contain 4 chromosomes. We'll have 2, a large and a small, that would represent a single genome. Those 2 in blue we can say are from the father. And then we'll also have a large and a small in red that would have come from mom. This is now a diploid cell that contains 4 chromosomes. The 2 long chromosomes would be an example of a homologous pair. And the 2 short chromosomes would be a homologous pair. The first step in the cell cycle is going to be to bring this cell through the stages of interphase, specifically S phase. It is in S phase where all of the DNA in the nucleus will replicate to form sister chromatids. We represent our sister chromatids as these connected X structures. 2 from the father, and again 2 from the mother. The cell will now enter mitosis, where prophase will begin. In prophase, the nuclear envelope will start to disappear, so there's no more nucleus in our cell, and the mitotic spindles are going to start to form from the centromeres. In this cell, we still have the sister chromatids, and a few of those sister chromatids may have started to attach to the mitotic spindles that are leaving the centrosomes. The mitotic spindles will attach to the sister chromatids at the centromere. Associated with the centromeres are 2 protein complexes called the kinetochores, one on each side of the centromere associated with the individual sister chromatids. It is the kinetochore that forms the attachment site for the mitotic spindles. This arrangement ensures that each sister chromatid is attached to a microtubule radiating from one end of the cell. The cell from prophase will now continue through prometaphase and into metaphase. During this process, one microtubule from each side will have bound to the kinetochores of a sister chromatid and pulled on those chromatids until they are all lined up in the center of the cell, as shown here. And we can see the microtubules bound to those sister chromatids extending from the centrosomes. There is a protein holding the 2 sister chromatids together, which is degraded, causing the sister chromatids to separate from each other as the cell moves into anaphase. During anaphase, we will see that the sister chromatids have separated and are now going to be pulled to opposite ends of the cell by the mitotic spindles that they are attached to. The final stages of telophase and cytokinesis will then occur to divide the cell into 2. In telophase, the nucleus reforms, and in cytokinesis, we've now separated the cytoplasm from each other. Each one of our daughter cells will now contain the individual chromosomes that were separated during anaphase. These 2 daughter cells are genetically identical to the original. Both contain 4 chromosomes, 2 red and 2 blue. As mitosis ends, cytokinesis begins and the parent cell divides into 2 daughter cells. In animal cells, this stage begins when a ring of actin filaments, called the contractile ring, forms along the cell membrane. This ring contracts, pinching the cytoplasm of the cell, eventually dividing it into 2. In plant cells, cytokinesis must work differently because of the plant cell wall. Here, the division of the cell is achieved by constructing a new plant cell wall along the cell plate.